Namaskar. Today we are going to study about competitive ELISA. So you can refer to our earlier classes on ELISA, types of ELISA. There are 4 types of ELISA: direct ELISA, indirect ELISA, sandwich ELISA. All these you can refer to our previous classes. Today we are going to study on the competitive ELISA. When we say competitive ELISA means, suppose say there is antigen, then the antibody in the sample can bind to this antigen. We can add another antibody conjugated with enzyme. So the antibody in the sample and the antibody conjugated with the enzyme both will compete for the same antigen. Who will win? Whose concentration is more? If the antibody in the sample is more, then that will bind to the antigen. If the antibody in the sample is less, then the antibody conjugated with the enzyme will bind. And if the antibody conjugated with the enzyme binds, then there will be change in color if we add the substrate. So we have already discussed what type of enzymes we use, what type of substrates we use, and what are the color changes. So please refer to our earlier classes. So competitive ELISA, basically we can use it for both detection of antigen and antibodies. Mostly it is used for detection of antibody, for quantitative analysis. So both qualitative and quantitative we can do. For quantitative analysis particularly competitive ELISA is used. And this is basically used when the antigen cannot be sandwiched between 2 antibodies. In that case, we use this competitive ELISA. So what is that competitive ELISA? Suppose let us discuss for antibody detection. That means the sample contains the antibody and we want to detect that. So we'll take one antigen specific to the antibody and we will coat that on the microtiter plate. So if our sample antibody is if our sample contains the antibody, then that will bind to this antigen. And if our sample does not contain antibody, then it will not bind. So if we have added another antibody which is conjugated with the enzyme, that means there will be competition between the antibody present in the sample and antibody conjugated with the enzyme for the specific antigen. So if sample has more antibodies, then the antibody in the sample will bind to this. There will be no binding of the antibody conjugated with enzyme. If our sample has less antibodies, then in the competition, whose concentration more? The concentration of antibody conjugated with enzyme will be more. So that will bind instead of the antibody in the sample. So if sample antibody is more, antibody in the sample is more, then that will bind. If antibody in the sample is less or antibody conjugated with enzyme is more, then that will bind. So this is the competition. That's why you call it as competitive ELISA. So we'll take a ready-made microtiter plate coated with the antigen, blocked with the BSA. Why blocking is needed? Why washing with PBS is needed? That part you can refer to our class on ELISA. Then we'll add 2 antibodies. One, the sample or serum in which we want to detect the presence of antibody, and another enzyme conjugated antibody which is also specific to that antigen, which is coated on the ready-made microtiter plate. Suppose say we want to detect the presence of HIV. So the ready-made microtiter plate will be coated with HIV. Now if the patient is HIV positive, that means he has developed antibodies against HIV. That means HIV antibody is there. and we will add another antibody which is conjugated with the enzyme. Okay, so now there will be competition between this and this. So if the person is HIV positive, that means this antibody is available, then it will bind. If the patient is HIV negative, that means this antibody is not there, so this will bind. If this binds, then there will be change in color when we add the substrate. If this binds, that means this does not bind, so there will be no change in color. This is the principle. So then we will have washing. Now the competition between the sample antibody and the enzyme conjugated antibody will be there. And if, that means there can be two possibilities: antigen binding with the sample antibody or antigen binding with the enzyme conjugated antibody. So if the person is positive, then antigen will bind with the sample antibody. If the person is negative, that means there will be no antigen sample antibody binding, there will be antigen enzyme conjugated antibody binding. So when there is competition, the concentration, the substance whose concentration is more, that will win in the competition. So when a person is HIV positive, that means HIV antibody against the HIV is more in the person. So in that case it will bind. If the person is HIV negative, then this antibody against HIV is less or not available. Then what we have added, the enzyme conjugated antibody, that will bind. So there will be change in color. We can detect the antigen also. So the same thing, microtiter plate coated with capture antibody, washing with PBS, blocking with BSA or non-fat dry milk as the blocking agent, washing, add sample antigen and enzyme tagged antigen. So we call here it as enzyme tagged antigen. Now the competition will be there. If sample antigen is more, sample antigen antibody binding will be there. If enzyme tagged antigen is more, that means sample or sample antigen is less or nil, not available. Then in that case enzyme tagged antigen and antibody will bind. Then washing, add substrate, color change, color intensity is more, then enzyme tagged antigen binding, that means sample is negative, antigen is absent or less, color intensity low, sample antigen binding, sample is positive, antigen present or more. So from the color intensity or optical density we can have the interpretation from the standard curve. Using competitive ELISA we can also do the quantitative analysis. Usually with increase in concentration, the absorption or optical density increases as per the Beer-Lambert's law. But here we just get an opposite relation. The curve looks like this. Why? Because if the target protein, say antigen, is more, that means the antigen is binding and the binding of the enzyme labeled antigen is less, and if enzyme labeled antigen is less, that means the color intensity will be less and optical density will be less. Whereas in case of low level of the target protein, say low antigen, then higher binding of the enzyme labeled antigen and there will be high intensity of the color, and because of that the optical density will be higher. So this is for the zero level of the antigen and this is for the high level of antigen. The optical density is less. So with increase in antigen level, the optical density decreases, and with low level of our target protein or antigen, the optical density is high. Thank you.